Hi, let's study about complementary gene interaction today. So what is meant by complementary gene interaction? It's a gene interaction where two genes complement each other in the formation of a particular trait. In other words, a particular character is controlled by two genes which are located in two different loci, and these two genes are complementary to each other. Okay, that means here the word spelling is Complementary. It's a term that is produced from a Latin term that denotes complete, not the other complementary, that is COMPLI. Okay, just understand that. So here in complementary gene interaction, the expression is complete only when the dominant alleles of two different genes are present. I will explain with the help of an example. In sweet pea, that is in lathyrus odoratus, flower color is controlled by two complementary genes. So these two complementary genes are present in two different loci, but these two are complementary to each other, right? Suppose this is one gene, the dominant allele of one gene. This is the dominant allele of the other gene. We can say it as capitals C gene and P gene. Okay. So C and P, these two genes are complementary to each other for the production of flower color. Okay. So for the formation of anthocyanin pigment, both the genes C and P are necessary, and also in at least one dominant allele is necessary of both the genes. You just understand well that at least one dominant allele of both the genes C and P are mandatory for the formation of purple flower in lathyrus odoratus. Why is it so? You know that this purple flower color is due to the presence of anthocyanin pigment. And this anthocyanin pigment is produced from a precursor called chromogen, right? So chromogen is produced only when dominant allele of C gene is present. So this C gene is responsible for the formation of chromogen, and chromogen is converted to anthocyanin by the dominant allele of P gene. So that is why these two are essential for the formation of anthocyanin pigment. Okay, suppose this gene is absent and only the recessive alleles of P gene are present. What will happen? Chromogen is produced, but there is no dominant allele P for the conversion of chromogen to anthocyanin. So the flower color remains white. What will be the case if C is capital C or dominant allele of C gene is absent? Chromogen is not at all formed from the precursor. Okay, so chromogen is not formed. If chromogen is not formed, there is no question of anthocyanin formation. So the flower color remains white only. If both the genes are in recessive condition, or suppose dominant alleles of both the genes are absent, then also chromogen is not produced, anthocyanin is not produced. So neither chromogen nor anthocyanin produced, right? So then also the flower becomes white only. Hope you understood. Okay. So suppose two white plants are crossed. See, capital C, small p. This is the genotype of first white plant. It is white because capital C allele or dominant allele of C gene is present, but dominant allele of P gene is absent. Now you have got the idea, right? So here it is white in color. The next parent is small c, capital P. Can you see it well? So here both the dominant alleles of P gene is present, but recessive alleles of C is present along with. Okay, right. So these two are white flowered plants. See, So these are the white flowered plants, genotype of white flowered plants. Now let's see which are the gametes formed from this. What's the gamete which can be produced from this capital C, small p? You know it well. So this is a gamete that can be produced from this capital C, small p. What about the other one? Here or here, small c, capital P. What will be the F1 genotype? Capital C, small c, capital P, small p. Is it okay? So here the F1 genotype is capital C, capital P, small p. Here you can see the dominant allele of both the genes are present. So what will be the phenotype of F1? All the individuals F1 offspring are purple flowered because dominant alleles of C and P are present. Now, what are the gametes which can be produced from this F1 individual? This capital C can fuse with either capital P or small p. This can fuse with either capital P or small p, right? So, capital C, capital P, capital C, small p, small c, capital P, small c, small p. These are the 4 gametes that can be produced from this F1 individual. Now, let's see how the F2 ratio comes out. Okay. So, before passing on to the F2 offspring, let's see once again how the F1 individual is produced. Here, this is about the complementary gene interaction. The flower color in lathyrus odoratus is an example that we are discussing. These two are the two parents. Both are white phenotypes. And the genotype you can see here capital C, small p. Only the dominant allele of C gene is present, so it is white. Here in small c, capital P, it is also white because dominant allele of C is absent. And one thing that you have to remember is be very careful while writing capital C, capital P and small p because it can be mistaken easily. Capital P and small p, capital C and small c. So be very careful while writing these lowercase and uppercase letters. Now in the first individual you can see this is the gamete that can be produced from the first parent and this is the gamete that can be produced from the second parent. And this is the F1 phenotype and genotype. Capital C, capital P, small p. Purple flowered plants. So all are purple flowered. In F1, all the F1 individuals are purple flowered. And as I showed you earlier, these are the gametes which are produced. So on selfing, the F2 individuals are produced like this. You can see the Punnett square. Here, see the first gene is, first gamete is capital C, capital P. Here also capital C, capital P. The genotype produced here is capital C and capital P. Here all the 4 dominant alleles are present. So it is purple in color. So anyway, I am not explaining all these things because now you have understood how the phenotype comes considering each and every genotype. Okay. So see here how many purple flowered plants are produced. You just count it. 9 offspring are having purple flower color and 1, 2, 3, 4, 5, 6, 7. 7 are having white flowered phenotype. Okay. So the complementary gene interaction ratio becomes 9:7. Here the F2 Mendelian ratio 9:3:3:1 is modified to 9:7. Okay. So hope you understood. So to conclude, in complementary gene interaction, a particular character is controlled by 2 genes which are located in 2 different loci, and these 2 genes are complementary to each other. That means at least one dominant allele of both the genes are mandatory for the formation of a particular trait. And also here, the 9:3:3:1, the F2 Mendelian ratio is modified to 9:7. Hope you understood. Okay, thank you.